Hello and welcome back, future scientists. I'm Mr. K with Mississippi Public Broadcasting, and I wanted to give you a first-hand look on how to solve some of these problems dealing with pedigrees and dihybrid crosses. I'm going to walk through a worksheet with y'all, and then that way when you've got some work to do on your own, you kind of know how to tackle them and can handle them on your own a little bit. So thank y'all so much for making it today. We'll go ahead and hop into this worksheet and see if we can figure out what's going on here. Okay, so we're dealing with pedigrees and dihybrid crosses. Okay, the first thing I wanted to do is kind of label this pedigree a little bit because we're going to need this to answer some of the questions. So the way we write this is, remember that each line is a generation. So we would write this as the first generation, this is the second generation, and then this line is the third generation. From there, every individual is numbered 1, and then going up numerically from there for a line, and we always do that left to right reading order. So we would say that this is individual 1, 1. This is individual 1, 2. So let me kind of go ahead and fill all these in, and you'll see why we're using these here in just a little bit. Okay? So again, each generation gets a Roman numeral, and then each individual gets a number under them, and you just start with 1 and you count up. So 1, 2, this one had the most, and so that's why we went all the way to 6. Okay, great. So let's go ahead and look at the questions and see if we can figure out what's going on. Number 1 says, how many generations are in this pedigree? Well, we can see here that there's 1, 2, 3 generations. Okay, so that'll be the answer to number 1. All right, number 2 says, how many individuals have the trait that we are tracking? Okay, does anyone know just by looking at this picture how many individuals have the trait? Well, this is going to be 3 as well. Remember that for whatever trait we're tracking, we designate that person as having it by shading in the shape, shading it in. So there are 3 shapes that are shaded, and that means that these 3 people have the trait. Whatever that trait is, these 3 people have it, okay? Number 3 says, how many males are in this family? Okay, what should we count to figure out how many males there are? What shape are we counting? We're going to count the squares, right? Because squares are males. So 1, 2, 3, 4, 5, 6. So there's 6 males. All right, how many females? Females are designated by a circle. So let's just count how many circles we have, and that'll tell us how many females are in this family. 1, 2, 3, 4, 5, 6. So 6 and 6. Great. Okay. Number 5 says, is the trait we are tracking dominant or recessive? This should usually be one of the first questions you ask yourself when you start trying to figure out all the genotypes, because that's really going to help us answer this question. Okay, if it's a dominant trait, it shows up in every generation. If it's not a dominant trait, it does not show up in every generation. In the first generation, there's nobody who has this trait, so this trait has to be recessive. Okay, this is a recessive trait because it skips a generation. Okay, so now what we're doing is we're going to go through and find out the genotypes for all of these individuals, okay, for number 6. Okay, so what we should do first, because we know that this is a recessive trait, we know what the genotypes are for all these shaded individuals. If they're shaded, they have this recessive trait, and they would only be showing the recessive trait if they were homozygous recessive. So we know that these individuals are showing the recessive trait, and so they have to be homozygous recessive, 2 little letters, okay? Then let's go through, now that we have that figured out, let's see if we can solve the ones that they're actually asking for. All right, so this first one says 1, 1. Okay, so they want to know what the genotype for this individual is. Okay, well, all the ones that are not shaded, the not shaded shapes here, we really don't know what their genotype is yet. This is where it kind of gets like a puzzle. You got to figure it out. We do know what we know what one of their alleles are. Because they don't show the trait, that means that they have to have at least one dominant letter. They could have both of them, but they have to have at least one dominant allele, otherwise they would be showing the trait. So what I'm going to do next is just go through and give everyone who's not shaded one capital letter. We know they have at least one capital letter, because if they didn't, they would be shaded in. So we can determine that. So here's where it gets a little confusing. It wants to know what this individual's genotype is. We're not really 100% sure right now. We know that they have to have one capital letter. What's that other letter? Well, remember, there's a very important rule I need you to remember. Your genes come from your parents. You get half of your DNA from mom, half of your DNA from your dad. So like, let's look at this individual. Where did these letters come from? Well, these letters had to come from their parents, right? And that means that one of the little r's came from dad and one of the little r's came from mom, right? So both of them are going to be heterozygous. That's the only way that they could have offspring that have this recessive trait. They must both be carriers. That's what we would call them, carriers. They carry the trait, but they don't show it. So both of the parents here are carriers, and that's how they're generating offspring that have this trait. So 1-1 is heterozygous. The next one, B, is 1-2. Same thing. We know that the dad here has to be heterozygous as well. All right, the next one is 2-2. So this individual right here, it's the shaded square. We know that that individual is probably, or has to be, homozygous recessive because they're showing the trait. So this individual right here, same thing. We know that this individual is shaded. They are showing a recessive trait, and that means they have to be homozygous recessive. All right, the next one is 3-1. All right, so it wants to know what this, they're asking what this individual's genotype is. Okay, this is kind of confusing, right? Because we know what one letter is, but we're not totally sure what that other letter is, right? Well, again, what you have to remember is that half of your DNA comes from your mom, half of your DNA comes from your dad. Where did this capital R come from? Well, it had to come from the mom, because the dad does not have a capital R to give. He's homozygous recessive. So that means that this capital R came from the mom. The only thing that dad has to give is a lowercase letter, right? So that means that this individual, 3-1, has to be heterozygous as well, okay? And then the last one, 3-3, okay, this is the other shaded square. Because it's shaded, we know that they are homozygous recessive. Okay, so that's kind of how you have to think about them. Go ahead, you want to figure out what the inheritance pattern is first. Is it dominant or recessive? Once you have that figured out, you can determine what the shaded shapes are. Especially if it's recessive like this, we knew right away that all of these shaded shapes had to be homozygous recessive. Then it's kind of like a puzzle. You got to look at the parents, what can the parent give you, and you can use that to determine what the other genotypes are. Okay, I've got another one down here for practice. So let's see. Okay, so 7 says, how many individuals have the trait that we are tracking? Again, we're just counting the shaded shapes. 1, 2, 3, 4, 5. I see 5 shaded shapes, so 5 individuals have the trait that we are tracking. Okay, 8 says, is the trait we are tracking dominant or recessive? Okay, well, does it show up in every generation? It sure does. And so that means that this has to be a dominant trait. Okay? I'm going to go ahead and label these for us real quick. Alrighty. So is the trait we're tracking dominant or recessive? It shows up in every generation, so that means that we're tracking a dominant trait here. Great. All right, so now it wants us to find the genotypes of these individuals. Let's just go ahead and start filling this in a little bit, though, and we'll get to those questions down here in just a second. Okay, so this one is interesting. The trait that we are tracking is dominant. So that means that the shaded individuals are showing that trait. All the non-shaded ones are not showing the trait. Well, think back to it. If this is a dominant trait that we're tracking, there's really only one combination of alleles that would not show it. So think about it like this. Only one of these three combinations over here on the side would not show a dominant trait, right? And it's this one. Because if you have one dominant allele, you're showing this trait here. All the non-shaded shapes have to be homozygous recessive. That's the only way that they would not be showing this trait. So let's go ahead and fill that in first. So we know all of these individuals that are not shaded are homozygous recessive. And we know that because if they had even one dominant allele, they would be showing this trait. Okay? So now that we have that figured out, again with the shaded shapes, we're determining if they're this one or this one. Okay? And we're not really sure, but we know that they do have at least one dominant R because that's why they're showing this trait here. So I'll go ahead and fill that in. We know that they have to have at least one dominant, and that's why they're showing the trait. All right, so from there, we're kind of getting it started. Let's look at these questions down here and figure out the ones they're asking for. All right, so the first one here says 1-1, okay? So what's the genotype of this individual right here? Well, they have the trait, so we know that they have at least one capital R. What is that other letter? Well, let's look at their offspring and see if we can get a hint there. And specifically, this is the telling one, right? Because this one is homozygous recessive, they've got two little r's. Half of your DNA comes from your mom, and half of your DNA comes from your dad. We know that dad only has a little r to give. So one of the little r's came from dad. Where'd that other little r come from though? Well, that little r had to come from mom, right? Because half comes from dad and half comes from mom, and that means that the mom here is heterozygous, okay? Great, so the next one is 1-2. So what's the genotype of the father over here? Well, they're not shaded, so we know that they have to be homozygous recessive. All right, C is asking for 2-2. So what's the genotype of this individual? Again, we're really not sure right now. We know they've got one capital R because they're showing a dominant trait, so they have to have at least one. Let's look to their offspring and see if that's a hint. Okay, so let's look at the daughter here. Well, the daughter is homozygous recessive, and that means that one R came from mom. Where'd the other R come from? It can only come from dad, right? And so that means that the dad here is also heterozygous, okay? All right, let's go to D. D is 2-4. We already have this one filled in. Because they are not showing the trait, that means that they're homozygous recessive. That's the only way you wouldn't be showing a dominant trait here. All right, the next one is 3-1. Same thing. We know that they have to be homozygous recessive. All right, and then the last one on this page is 3-3. Same thing here. We know that they are homozygous recessive because they're not showing the dominant trait. So that's kind of how you got to think about them. You can usually figure out one letter just looking at if the shape is shaded or not, but then sometimes to figure out that other letter, you got to go back and look at the parents or the offspring and ask yourself who's giving what, right? So you really just got to remember half of your DNA comes from mom, half comes from dad. They can only have one allele, and that can help kind of narrow down what the genotypes are. All right, so I think now, okay, I've got one more pedigree, and then we're going to do that dihybrid cross. Okay, so let's look at what's going on here. Okay, so 10 says, how many individuals have the trait that we are tracking? How many shaded shapes do we see? 1, 2, 3, 4. I see 4 shaded shapes, and that means that 4 individuals have this trait. Number 11 says, could this trait that we're tracking be sex-linked? Okay, for sex-linked traits, ask yourself, is it only affecting men? If it only affects men, it probably is a sex-linked trait. Only squares have this, so yes, this could be a sex-linked trait. Only males have this trait. All right, so now what it wants us to do is start filling in the genotypes of this individuals. Because we think that this is a sex-linked trait, remember that we're using X's and Y's here to determine those are the sex chromosomes. So we know all the circles are going to be XX. I'm going to go ahead and fill that in. All right, and all the males are going to be XY. And I'll go ahead and fill in this information as well. Great. All right, so now that we have that information, okay, so let's go back and kind of start filling it in before we were even looking at the questions down here. Okay, because this is a sex-linked trait, that's why we're using the X's and the Y's. Okay, but we also know that this is most likely, because it's sex-linked, all the men that have this trait have that recessive allele. So this one has the recessive allele, this one has the recessive allele, this one have the recessive allele. This is the only square that is not shaded in, and so that means they have a dominant allele there. Okay, so now let's see if we can kind of figure out what's going on. Okay, so number A says 1-1. So we want to know what the genotype for this individual is. Okay, so here's what's going on. Let's look at one of their, again, we're not really sure what goes over the X's, right? But we can kind of figure it out. So if we're looking at this, we can figure it out, because when you're looking at the boys here, their sons, the Y is coming from dad, right? The mom can't give a Y. So that means that both of these Y's are coming from dad. And that tells us what one of the alleles are. Because both of them have the X little b, that X little b is coming from mom, because she can only give an X. Okay, so we know she has at least one lowercase letter, and we know that she has one uppercase letter, because if they were both recessive, she would have this trait. Okay, so I know that one's a little confusing, but here's the genotype for the mother here, is she would be heterozygous for this trait. All right, let's look at the next one, 1-2. Okay, so for this one, they're asking about this individual. I'm going to erase some of this so y'all can see what I'm talking about a little bit better. So this individual right here, this is a man. So we know he's XY. He has this trait, and that means that he's got to have that recessive letter there. All right, the next one, C, is 2-2. So this individual right here, same thing as the dad. It's a man, so it's XY. He has the trait that we're tracking, so he's got that recessive letter. Okay. The next one, D, is asking about 2-4. So this individual. Okay, let's see if we can figure out what her genotype is. Okay, well, she is not showing the trait, so we know that she has at least one capital letter. She's a female, so she's got two X's. What is going to go over this next X here? Well, let's kind of think about it. She got the X capital B from mom, and that means she must have gotten the X lowercase b from dad, because that's the only X that the dad has to give. So that means that this is her genotype. Okay. All right, let's keep it going. E says 3-1. Okay, so 3-1, what's the genotype of this individual? Okay, well, it's a male, so it's XY. He is not showing the trait, so he's got a capital B. Okay, that's the only way a man would not be showing the sex-linked trait, is if it's that dominant one. All right, and then the last one here is 3-3. We already have this one figured out because it's a shaded male. We know that it's XY, and that on the X it's that lowercase letter. Okay, so again, very confusing, especially when you're getting to the sex-linked stuff, but just kind of use some of those steps that I gave you, and hopefully that'll make those a little bit easier. All right, y'all, so this is a dihybrid cross that we're going to do, okay? You can see here the Punnett square is a lot bigger than it normally is. There's 16 cells instead of just the 4. Okay, but the first thing we have to do is to figure out what the gametes are. And what I want to do is use something called the FOIL method, okay? And that stands for first, outer, inner, last. We're going to use that to determine what the gametes are here, okay? So it says, what are the gametes for the first individual? So let's look at this genotype, and we're going to figure out what the gametes are here. So we're going to take the first of each allele combination. So that's a capital A and a capital B. This is the first out of those two, and that's the first out of those two. So this is one gamete that they can produce, okay? Then we'll go to outer, okay? What are the 2 alleles on the outside? This one's on the outside, this one's on the outside. So another allele they can make is capital A, little b. All right, then we'll take the 2 on the inside, okay? These are the 2 on the inside. That's a capital A and a capital B, okay? And then the last out of each combination. This is the last one. Let's put a check mark by it, and then that little b is the other one. So another allele combination that this individual can make would be like this. Okay, so that's the alleles for one parent. Let's see if we can find the gametes for the other parent. Okay, so now let's look at this one. And again, we're treating the 2 A's, that deals with one trait, and the 2 B's deal with one trait. We're going to FOIL this genotype out to figure out what the gametes are. So the F stands for first. We're going to take the first allele of each trait. So that would be the capital A and the little b. That's the first one out of those pairings. So A, little b. Then we'll do outer. Which 2 letters are on the outside? This one's on the outside, this one's on the outside. So it would be capital A, little b again. All right? And then we'll take the 2 on the inside. I stands for inside. So which 2 are on the inside? It's these 2. Okay? So that's a little a and a little b. And then the last of those 2. So this one is the last, that one is the last, and so that's a little a and a little b. Okay? So now what we'll do is use that to fill in our top and side of the Punnett squares, and then we'll have to fill everything else in. So here's the gametes for one parent. Okay, here's the gametes for the other parent. Okay, when I'm doing these, I like to just focus on one letter at a time. It makes it easier for me to see what's going on. So I'm going to go through and fill in all the boxes with the A's, and then I'll come back and fill in all the boxes with the B's. Okay, that just makes it easier on me, but you do whatever you think is the best way to get this done. All right, so I filled in all the A's. I'm going to go back and do all the B's next. Notice how I'm keeping the similar letters together. So both of the A's go together, and then both of the B's go together. And then within that, we're still keeping the dominant letters first and the recessive letters second, okay? It's very easy to make a mistake, so take your time. All right, so now that we've got the dihybrid cross filled in, we can start answering the questions here. So the question for this one is, what is the genotypic ratio for the offspring. So what I want to do is go through and find all of the combinations up here that are similar. We're going to start off with this one. So 2 capital A's, 1 capital B, and a lowercase b. Let's see how many combinations of that we can find. There's one here and there's one here, and that's it. Okay, let's go to the next combination. So 2 capital A's and 2 lowercase b's. Every box that have that, I'm going to put a little check mark by it, and that's going to help me figure out the ones that are similar. So that's one, and then that's the other one right there. All right, let's see if we can find another combination. Okay. So what we should do here is all right, so when I was foiling this second one, I made a mistake. So you can see here that for this individual first, outer, no, that's right. Okay, so let's keep this going. Let's see if we can find all these combinations that are alike. Okay, so here are some that are similar. So the 2 capital A's, 2 lowercase b's, those are the same, those are the same. Here's another combination like that. Okay, so let's go to this next one then. 2 capital A's, 1 lowercase b, and a lowercase b. All right, so those are, 4 of those are similar. Let's see if we can find any more like that. Okay, we're good on that. Okay, so the next combination, let's do heterozygous for each. You see a capital A, lowercase b, uppercase B, lowercase b. Let's see if we can find all the combinations that are like that. I'll circle these. Okay, so that one. Okay, this one. Okay, so those 4 are all alike. And then our last combination, I will put an X by these. Uppercase A, lowercase b, all 4 of these are alike. Okay, so the reason I was going and circling them and underlining them and check marking them is it's going to make answering 15 a little bit easier. So I'm going to look at all the ones that are underlined right now. We have 4 that are underlined, and the underlined ones are all like this. Okay? Okay, what's the next combination we have? Let's look at all the ones that have check marks by it. So we have 4 that are like this. Okay? Let's look at the ones that are circled. Okay? There are 4 that are like this. Alrighty? And then the ones with X's next to them, there's 4, and they're all like this. Okay? You know you've probably done them correctly. You've at least found all the correct combinations, is if when you add all of them up, if they equal 16. So 4, 8, 12, 16. That means every box is accounted for. We might have still made a mistake if we did our foiling incorrectly. I thought I had done it wrong there for a second, but once I went back and double checked it, I think we're all good there. So here's the genotypic ratio for the offspring. It's 4 to 4 to 4 to 4, and you could simplify that as 1 to 1 to 1 to 1. But for now, that'll help us know that every box is accounted for because it adds up to 16. Okay, y'all, thank you so much for following along with me today. I wanted to make sure I did those with y'all because you can see how complicated they are. It's very easy to make a mistake. So take your time, use those steps that I gave you, and that's going to make completing these easier as you move forward. Okay, your teacher has some other work for you to practice this stuff on. Make sure to give that your best effort, and I hope everybody has a great day.